DNA, deoxyribonucleic acid, is an information molecule that contains genetic material and provides instructions for making proteins. DNA replication refers to a process of taking the original DNA and making it into two new DNA, starting at a specific base sequence location called the origin of replication. Since it does not affect the other side at all even if something goes wrong on one side, this replication is considered to be safe. To begin with, DNA replication begins at the initiation point when the helicase breaks down hydrogen bonds. This process allows double-stranded DNA molecules to decompress into two single strands, allowing each strand to replicate. Next, topoisomerase loosens DNA. Topoisomerase is a ring-shaped protein that allows DNA to maintain its spiral structure and prevents DNA strands from getting tangled. DNA strands can be unwound or rewound using topoisomerase. These loose DNA strands are separated by single-stranded binding proteins that prevent DNA strands from sticking again. At the end of these steps, primase adds an RNA primer to the DNA strand 3' direction and creates a new DNA strand 5' direction. The primer consists of 5 to 10 RNA nucleotides and serves as an initiation point for additional DNA nucleotides. In this strand, in the direction of branch replication point 3', the primer is added only to the end of the strand. On the other hand, in the strand in the direction of branch replication point 5', the primers are added to several points. At this time, the polymerase adds nucleotides to the 3' end of the DNA strand. The reason it is added only to the 3' side is that DNA is bound into the 5' to 3' direction. And then, the leading and lagging strands are formed. Leading strand refers to an ever-forming strand of DNA, while the lagging strand is a fragmented strand of DNA. DNA polymerase enters to remove RNA primers and add supplemental DNA nucleotides as the next step. Then the ligase connects the empty space between the DNA strands. After all these processes, two DNA double helix structures are finally created.